mentioned. Hopefully my computer is acting a little strange this morning. This will work okay. Okay. We're going to talk about the Cancer Genome Atlas, publicly available data, and how you could use this in your research. So a little bit of background about the TCGA project. This is a large-scale collaborative effort that was spearheaded by the NIH a few years back with a pilot project looking at glioblastoma samples. The idea is to create a comprehensive map of the genomic changes in different types of cancers. After the initial pilot project, the goal was expanded with additional funding from the Recovery Act, and the current goal is to examine over 11,000 samples across 20 different cancer types. One of the major objectives or the goals of the TCGA project has been to disseminate the data information as fast as it gets generated through a public access database repository. And they've done an excellent job at that, and the data is available at different levels. We'll talk about that. And the ultimate goal, of course, is to improve cancer diagnosis, treatment, and prevention. All this data actually comes from their website. So if you go to the TCGA website as of a couple of days ago, you can find a list of the different cancer types, like the AML and bladder, etc., and how many samples that they have collected and how many of them are ready for being downloaded at different levels. What I mean download, this is just taking big files down. And so you can see there's quite a bit of cancers and quite a bit of samples available. Now, what are the different types of data that is being collected? So first, there are, for each sample, typically, in all the cases that I've seen, it includes the tumor, genomic profile of the tumor, the match surrounding normal tissue, and the blood-derived normal for the same individual. And samples were processed at multiple sites using different platforms. This was at least true for the pilot project with the GBM, glioblastoma, where the aCGH Agilent oligo arrays were used at a couple of sites, I think Harvard and Sloan Kettering, and then SNP arrays from Illumina and Affymetrix were used to profile the same samples at other institutions to look at if there's any biases or differences between different platforms. So there's comprehensive data there. In addition to the genomic profiles, there's also a lot of phenotypic, well, some phenotypic data for each sample, and it depends, the extent of that, it really depends on the different types of samples. But most often, all the ones that I've seen include survival data, which is also very nice to have for doing biomarker discovery type of research. Now, along another dimension, there is of the genomic data, there's, in addition to copy number, there's gene expression, methylation, mutation, LOH, data that's being measured in raw format to get at. So how do you access this data? So what TCGA has defined, or NIH, or NCBI, whoever is the authority up there, has defined 4 levels of access. There is level 1, which is raw data, and we're going to concentrate on copy number. So with regards to copy number, level 1 data is probe measurement. So in the case of Affymetrix array, it's the cell files. Level 2 data is, so level 1 is unnormalized data. Level 2's idea there is once some normalization happens at the single sample, at the single probe level. One thing that, at least for me, is a very big challenge is navigating the data that the NIH generates and puts on the website. It's actually rather difficult. You need to spend quite a bit of time. I have not been able to find level 2 data. Doesn't mean that it doesn't exist. It's just that for me, I haven't been able to find it. I'm not the expert there. But you can easily find level 3 data, which is segmented data for copy number. This means these are regions that have a gain, loss, or LOH type. And again, there's a mysterious level 4, which is some summary finding data out there. So what the access control that the NIH has put is level 1 to get access to the level 1 or raw data, or level 2 you need to have special permission and IRB approval, etc. to since there are information about SNPs and other things that could make these patients identifiable. So you cannot access that publicly, but level 3 and up are information that's publicly available. So regions of copy number change, segmented data, those are publicly available. And that's what we will talk about. So what we have done is downloaded and organized level 3 data. So this is publicly available data that you can get access to. So how useful is this data? How can it be? Is it really useful after more than $200 million spent on it? I think it's amazingly useful to help in discovery. The amazing resource for all our researchers. There's been already a couple of major publications, at least the GBM and ovarian cancer publication that came out of the earlier data sets, and there's more work that's being done on it. Unfortunately, I think if you look at what publications have come from it, mostly the groups that generate the data are more familiar with how to get the raw data and access it and organize it. The difficulty is, although the data is publicly available, it's rather complicated to organize it for proper mining and exploration, and that's where we're trying to help the research community. So what type of analysis can you do on this data? So one thing is very simple. You can issue queries based on genes or regions across different sample types. If I get a chance, we can just do that on the TCGA website. You can ask, for example, in all the GBM samples, what's the percentage of EGFR amplification, or do it across tumor types. But the real power, I think, of the data is in aggregate type of analysis. When you pull together and ask questions like, what are the common regions of gain and loss across all tumors? So are there oncogenes or verifying the oncogenes and tumor suppressors that span multiple tumor types? And then there are other things where you try to identify or differentiate between different types of tumors, do clustering, do other types of analysis. You would need to have more than just a simple region query. So the TCGA portal, right now, it allows you to ask very simple queries. Like I said, what percentage have EGFR amplification, but nothing very complicated from there beyond that. In addition, the data that's available, the level 3 data, has some issues, at least with regards to the copy number, that it doesn't make it the ideal. I think the underneath level 1 data, a free process might provide better insights. So those are some of the limitations of what's currently available on the TCGA website. So what we have done is we have tried to solve some of those problems. So at BioDiscovery, we have a local expert who has gone and downloaded all the level 3 samples and associated phenotype information and merge them together and create Nexus projects, one project for each different type of cancer, and then this has been loaded up on our Nexus DB repository, which is a cloud-based system. It sits on the Amazon AWS cloud. And Nexus DB can be accessed directly from the Nexus copy number tool. So if you have Nexus copy number, all you need to do is to just create an NexusDB account, and then you can query these samples at will, as well as download the samples and do further investigation, either merging them to the project that you have studying, or just start looking at the TCGA data. So that's my slide presentation. I usually stop to see if there are any questions at this stage, and I don't see any questions now. Shalini mentioned this is a one-way audio, so if you have any questions during the presentation or at any time, go ahead and type them in. At the end, I will stop and we can go over your questions. So this is a Nexus project that I have open. For those that are not familiar with Nexus, a project is just a collection of samples. So in this case, if you look on the corner lower left, over 1,000 individual rows or samples in this case. This is the glioblastoma set done with Agilent level 3 data. So each row shows the TCGA sample ID, how it was processed, and the data type says database processing because I did not process this data. This comes directly from NexusDB. So this has been all downloaded here. And all of these phenotypes, like histological type, days to death, barcodes, if it's a primary tumor or blood drive, these were all information that we gathered from the TCGA website and brought it down here. So if I select all of them to view together, actually maybe instead of looking at all of them together, let me sort based on the patient barcode and we can grab this one. So this is the same patient. This is the primary tumor. The next one is the blood-derived normal. So if I'm just interested in looking at a single sample, in that way I can click View. And let's just look at these this one sample. And one thing I noticed is, so this thing is red. This is a filter. So I did this for the webinar earlier. Let me remove the filter so the data is displayed with all the calls there. So this is the normal and this is the tumor. And if you're familiar with GBM, a hallmark of the cancer is trisomy on 7 and monosomy on 10. You don't see that in the normal. You do see quite a bit of gain and loss. The gains are blue and the red indicates loss. If I click on the normal, so this is the normal sample. Let me actually expand this completely out. So this is called a single sample drill down in Nexus. So you can see all the phenotype data that we have. This is a female patient still alive at the end of the study, and some of the other factors. The overview shows this is apparently what's in the germline, lots of changes. And now here's what level 3 data looks like. Well, I should have maybe taken the cancer, but this is the bars are the segments that's coming. So let me zoom in to an area. So these are all the segments that the pipeline, I think at the Broad or wherever the segmentation was done, chopped up the genome in these regions. And then we have defined if a gain as exceeding a threshold of 0.1, that would be a 1-copy gain. And if that bar goes below minus 0.15, it's a loss. And then there's amplification, which is this bar, and homozygous deletion, that bar. So based on that, we get all of these calls. And if you go to, for example, let's go to chromosome 2, and you can see there's like a lot of gain and losses. So they're saying that for this gene, that region was amplified. Now, well, you don't see the probes because that's not part of the level 3 data. Let me close this one and bring up the tumor because it's more informative. One of the the problems I think with the level 3 data, there's two things. One, in this case, actually, this might be fine. Sometimes you have the ploidy problem where the baseline gets shifted if you have lots of losses, and that's an artifact that needs to be corrected, and it's not really corrected in level 3. And also this over-segmentation. So you can see there's just amazing number of tiny calls. And in fact, if I sort based on the size, this is a 6 base region, or an 8 one basis. I think these are single probes, and they just don't make much sense. So our goal is actually, we're in the process of getting authorization to take level 1 data and reprocess them in Nexus, so we don't have that whole issue. But what I do for the analysis right now is I just remove everything that's less than 50 KB, just so it looks a little bit cleaner like that. So now this looks a lot better. This is normal, and some of these events, so if you look around here, you see this gain, that's a known area of copy number variation. There's 21 gains that were reported. So let's go back into the main data set and select all of them. So what I've done, this is, again, this data came from NexusDB, and I'll get to that in a second. Just want to show you, once you have it, what you can do with this data. I can view all the 1,000 samples together by clicking the view button. Now, as opposed to having just a pair, we see multiple samples down here. And up here, this is a frequency now. The frequency plot that makes more sense. So about 50% of the samples, for example, have this trisomy of 7, and about the same number have the deletion on 10. It's 50% because if you recall, I think sample type, yes, the orange ones are actually the primary tumor, and then there's some normal. So if I collapse this and create an aggregate for each sample type, the 578 primary tumors, they have, almost all of them have this gain, and now you can see the normals don't have that. They do have this funny CNV-looking thing around the centimere of 9. So this is, I can do a number of analyses at this point with the data, but let me now talk about how this data came to my computer. So if you go to if you're using Nexus copy number, you have this tab called NexusDB. If you go there, there's one login button. You click on that, and you can log into NexusDB. If you have not created an account, it's very easy. Anybody that has the software can create an account. So you just click on Register Account, put in your information, and you get an account. There is a bit of a misconception. So if your lab has a license, or your PI has Nexus copy number, everybody does not have to or should not have to use the same login account. The number is not fixed to the number of seats of the software you have, so you can have multiple people with their own account. That's important because, let me log in. So, because when you log in, if you ever upload your own project, so here, these are the projects that I've uploaded. They're private to yourself. Nobody else can see it. So even if they're colleagues in your lab, they won't be able to see it because it's registered to you. You have to create a group and allow that group access to see your project. So right now, what I want to do is to go to the visible projects. These are the projects that I don't own. Other people, these are the other owners that own these projects, but they have either made them public to anybody in NexusDB or a group that I belong to, so I can access them. So if I go down, this is, and you can see there's a bunch of data sets that we've downloaded from Geo, and like this one here is a breast cancer data set that was in Geo. and you can see the factors for it. But I want to go down to the TCGA section. So you can see here, now we have this set of projects. These are downloaded level 3 data on build 37. Some of them are quite large, like this ovarian cancer is 4,000 samples, over 4,000 samples. And then some are small, like this pancreatic cancer, because I think TCGA is just going to generate more. One thing to note is that, as you notice, when I click on it, it shows down here what are the phenotype data that's stored in NexusDB for that. So for this pancreatic cancer, this is really not a lot. But for this ovarian cancer, you can see that we have some information. Some of them have more, some less, like this one. The neck cancer has smoking history, information like that. So if you want to start mining the TCGA data yourself and getting access to it, you can create a project, and then in this case, you see there's this low-grade glioma project that's interesting. So I can click on that, and you see these are the 200 or 285 samples that uploaded in that project. And these are the phenotypes associated with each sample. Now, if I have this glioblastoma project in here, and I'm thinking of a research study that says, well, what's the difference between a high-grade tumor like that versus a low-grade glioma, I can go in here, I can sort based on sample type and say, I don't really care about the blood-derived normals, and I just want to take all the samples that are primary tumors and select them. So it says 144 got selected, and click download. And there's a tag. Sorry. The tag is there for you to use. I find it a very useful feature because you can query the database multiple times, and sometimes you forget why you downloaded the sample. Sometimes you do a query for, like, let's say EGFR amplification, find those samples that have that, and just want to mark them. And that's what that does. So what's going on right now is, it's going a little too fast. I was going to explain. What's happening is contacts the Amazon server for NexusDB. It has it zip up those samples. Then they get downloaded to my local machine, which is what happened a little bit ago. And then they get unzipped and added to the project. So it's a fairly efficient flow. For level 3 data, since it's not probe level data, as you can see, the process is quite fast. And I just got 144 samples in a matter of a minute. So theoretically, you can just go ahead and download all the samples that we have put in here from TCGA into your project in a matter of an hour or so, and have a few thousand samples to play with. So right now, this is just wrapping up and adding the samples into my project. And now I can proceed to compare gliomas, high grade and low grade together. So if you look now here, my total count of samples went to 1,242. So I added about 140 or so new samples. And if I sort on the Nexus tag, now you can see these are all the new samples that I've just downloaded, and with their annotations. This low-grade glioma actually has some better annotations, or some more interesting annotations. So what I want to do right now is actually I want to sort on the column says sample type and just select the primary tumor. So I'm going to select all the primary tumor samples, regardless of if they're GBM or lower grade tumors. And as soon as I get to the top, here, and I'll select those. So there are about there's 722 of these guys. Now, I have survival data for some of these, and I do have the histological type. So if I right-click and say, I wonder if I don't know much about this, there's 20 of them that don't have a value. So I can generate a Kaplan-Meier plot of that. And this is for the untreated GBMs. They have a very bad prognosis. The other green is the treated GBM, slightly better. Astrocytoma, oligoastrocytoma, and oligodendrogliomas, which is the lowest grade. So you can see that there is a nice separation between the histological subtypes and survival. And you can actually get the p-value, rank p-value for the differences between different groups here. So one question could be, yes, what's the difference between the GBMs and the oligodendrogliomas? And one way of doing that is you can visually do this. So if you were going to write a paper on that, I'm not sure if people have done that in the TCGA data or not, but if you wanted to, you could look one up fairly quickly by taking histological type and coloring each sample based on histological type, and then say factor aggregate, and then have a plot like this. There's 18 that don't have the histological, but then you can say, this is what the profile looks like for an untreated GBM, and this is for an astrocytoma, which, if you recall from the Kaplan-Meier plot, the survival was bad for both of these. And you can see the genomic profile looks very similar. There's some slight differences. So you can generate this type of questions. You can also do other things. So if I haven't done this, but you can go into the comparison. Actually, let me delete all of these things that I've done comparison before. If I wanted to ask that question, is that, well, there's astrocytoma and then the untreated, the primaries. What are the areas that seems to be fairly similar, but what are the areas of very significant difference? So I can go into the comparison and I can create a comparison based on histological types. And let's take for the baseline the untreated GBM. So here I have created multiple comparisons that I can issue, and I can take this one, which is the one that we were interested in, a bad one and versus a really bad condition, and click View. And it performs a Fisher exact test on regions of significant difference between the two populations. So even though they both have a gain between in chromosome 7, the frequency in this case is only 50%. In this case is almost 90%. So it's a very it's significant difference. And then some other points. So I'm going to change the p-value to 0.05 to make it 0.0001 to make it very stringent, and then loosen up the difference threshold to 15%. To see if there are other minor differences. So on chromosome 9, I bet you, so this is P16, CDKN2A, and the dark red here says homozygous deletion. So you can see that 68% of samples in GBM have a deletion of this tumor suppressor. It's not as bad in the 38 samples there. So that's one area that's different. There are other differences. There's something here which is very small. We can go back to, actually maybe let's issue it from here. Let's look at this tumor suppressor. And the question might be now, what about other samples, TCGA samples? Do they have P16 deletions? So there's this query database button. You click that, and then click at this point. What it's doing is querying now the database over at Amazon, as opposed for me to download all the 5,000 to 6,000 samples. But it's issuing a query out there and says, show me all the samples across these projects that have this T16, CTK, and 2A deletion. And it sorts them based on the frequency. So we can see that the highest frequency is in the GBM, and then next is in lung cancer, bladder cancer, etc. And it's very infrequent, apparently, in thyroid cancer, something that I know nothing about. So you can look at it this way. You can also get a graphical picture of those samples, how focal that event is. So if you see here, it's the whole chromosome 9, but let me zoom in a little bit and just go around. I can't grab this side. Anyway, so you can see. So I'm just trying to get to the focal, see if there's cancers that have a focal deletion like this one. This pancreatic cancer apparently has it. So if I go to, if pancreatic cancer is interesting to me, 6 out of the 26 have that deletion. If I click on the blue area, it brings up the 6 that had that deletion, and I can see the extent of it. So this one sample, sample 20, had a nice focal deletion at that point. And I could actually download those when I click on here. I can download these 6 samples and add them to my GBM project and continue with my research. Just want to show the fact that you can continue to mine the TCGA data without even downloading the samples. So that's one trajectory. And let me see how I'm doing on time. It's fine. Other types of analysis that you could do is once you have the data, if you're not familiar, I'll show you a few of the features. This is by no means exhaustive set of features in Nexus copy number. But here we can take from, let me do something. We're going to select the primary tumors of both cases. So let's grab all the glioblastoma because they're all primary tumors, and just to save time, I'm gonna just grab, if not too many, but a bunch of these GBMs as well. So I'm gonna select these guys and do a Kaplan-Meier plot just so we can see still the same thing. This one is the treated primary, which I didn't really want to have, but that's okay. So if I look at these primary tumors, and instead of separating them out based, okay, one thing we can do is the histological type and collapse that, as we saw before. So this is the profiles based on histology. But can we group them just based on their genomic profiles? Can we do a clustering? So we can run a hierarchical clustering on this set, and let's say we can divide this up into groups like that. I can add the histological type to see, like this blue area, for example, has a lot of this green, which is GBM. But then this part that's separate is astros and some other things. So you can divide this up into, let's say, these 9 categories based on the genomic profile and say, okay, cluster 9, just quickly. So what this does, if it goes back into the, let's go back into the data set tab once it's finished updating. It creates this new column, which is the actual clusters column. So this sample is an HC3 cluster, this one is an HC6, etc. So if I go and view the samples, and now as opposed to grouping them, actually before I do that, let's see if the survival is different between these clusters. Well, there's this one cluster, that HC1, that has one patient in it, and it's good. But if we look at the bigger clusters, HC3 has bad prognosis, and HC6 has better, and then we get some of these other cases. So if we want to look at that based on cluster and compact view, so you can see that this one was the bad outcome, and this one I think was better. So you can see this one doesn't have, for example, the difference between this cluster is it doesn't have the deletion of 10. And if you look at this 1P, that actually does go with a better survival. So you can look at visualize the data like this and mine the data. You can also ask the question, is what is significantly different between this one and that group, going through the comparison tab. There's a series of processes that you can do with this data set. So I don't want to cover all of the features, but maybe just to do this one real quick. Let's make this cluster 9 and compare cluster 6 with cluster 3. So if I want to do that, one easy way is just to pick cluster 3 as the baseline and just pick 6 versus 3 and view that. If you recall, I think this was the one that had a very big difference between the chromosome 7 and 10. And you can go into the regions tab. This shows you all those areas that are significantly different. You can click on one to see if there's any genes there. This one doesn't. Many of them don't have it, but this one's just a copy number. So let me highlight that and view the annotation. There's about 3,000 genes that are different between these two clusters in the regions of copy number change. And if I click the enrichment button, I can see what pathways are different. So the transcription, DNA dependent, biological processes. I can look at KEGG, biocarda. I don't know the MAPR pathway. And the ones that show up in yellow are those that are in regions of significant difference between the two populations. Again, I don't have time to go over the details of all the various exploration tools in Nexus, but want to give you a quick flavor of how you can download the TCGA data and make use out of it. So at this point, what I want to do is I see there's some questions. Hopefully I didn't confuse a lot of people, but we can go through and answer these questions. Please type them in if you have any questions, and we go from there. So the first question is for the level 3 copy number data from Affymetrix SNP6 platform on TCGA. There are, for each patient, the reference assembly, 2 files. One that has no CNV and the other one that has in the title, and one that doesn't. Is the file with no CNV in the title expression data. In the previous webinar, I mentioned you have to have your own PhD in exploration of TCGA website. I don't know. I'm not an expert in the website, and I was going to show the website and the nomenclature. But what I think they do have is that they do have processed the samples both as separate tumor and normal, as well as combined, so tumor minus normal. And if it's processed in that way, it does remove the CNVs. So the data that we have uploaded, I think for the most part, is separate. It's tumor and normal separate. What we are, I mentioned this at the beginning, what we are planning on doing is to process all the samples as matched tumor normals, and within Nexus, which I think would give better calling results, and then upload those, make them available on NexusDB as well. That's work in progress. Hopefully that answered your question. If not, please type it in again. Y chromosome shows 100% deletion. Can you explain? Well, I guess mostly were female. So this is the way this segment came from TCGA. So let me double check over here. Yeah, so the Y chromosome shows deletion in all of these guys. So if I click on one sample and look at the whole genome, let's go to chromosome Y. Yeah, so it's just the fact that they have a segment that's way off there. Just the way the segmentation called. You can filter out the sex chromosomes. I think one of the filters here is to remove the calls from the sex chromosome, so it cleans that up if you want it. So that's just an artifact of the segmentation. How will the logistic function work on these larger data sets? Oh, great question. So what TCGA, most of the publication has been done, I think one of the reasons the level 3 data looks like the way it does is most of the analysis has been done through logistic, which looks at the raw probe level data. Unfortunately, there is logistic built into Nexus, but you won't be able to run it on level 3 data. You need the level 1 data to run logistic on. And it would be very fun and interesting actually to run that and all the samples together. So that's something that we haven't done. The question is, will the slides be available later, the webinar? And the answer is yes. Thank you, Fad. Can you add NCBI Bill 37 data to a project? Excellent question. So this has to do with the build and the genomic build. So if you look up here, it says Nexus copy number, this project is Bill 37. So the data that I get has to map to this build. So when I go to NexusDB, you can see the build is over here. These are all Bill 37. Now, if I wanted to, let's take this set of samples, and so I haven't done this, so I don't know if it's going to break or not, but it should not allow me to, yeah. So it's not going to allow me because there's a mismatch. The project that I have is Bill 37, and this data came from Bill 36. So I cannot jump between the builds. So if you have Bill 36 data that you want to merge some TCGA data with, that's on 37, I suggest moving your 36 over to 37 and then downloading that. Okay. Hi, Ali. You mentioned the quality of level 3 number data and the algorithm they use to do this is questionable. Do you deem the data more? Oh, well, yeah, we talked about this. I'm not sure if that was at the beginning. So if you look at this sample, oh, maybe not that one, let's pick another one for sure. So you see where you see all of these gains and losses going on. And that's just because if you look at the whole genome, what happens is these deletions on these chromosomes have shifted up, these big deletions have shifted up the baseline up. So all of these guys have gone above the thresholds that we have for the gain. So you artificially, in some of these samples, you get some gains or it can be losses that are not necessarily true. One way the TCGA website gets away from that is move these thresholds farther up, which you can do, but then you would miss out on some mosaic events. So one problem with this level 3 is that. The other thing is just if I remove this filter, you end up with all of these over segmentation results that you end up with like tiny little deletions all over the place. So as I mentioned to you before, we are trying to, we have gotten the GBM set, and hopefully soon we can have the GBM processed in level 1 and then uploaded to the website. The question is, missed it in the event of a whole genome ploidy change, ploidy, how does, yeah. So I think we talked about that. The more specific question here is, how does Nexus find the proper baseline? So Nexus has two means. With TCGA, since there is a match normal, we can run aCGH on this, and we had a poster. If anybody is interested, I can send it out on the ovarian set that we ran aCGH on. aCGH is a great algorithm that you can run within Nexus to get you the ploidy and % contamination. Sometimes certain percentage of the samples, when you look at it, or when I've looked at it, I would not agree 100% with it. So I'm not sure if that's what we want to put in there. I think right now the best way is manual curation, and that's what we're planning on doing with the level 1 data. Question is, I'm concerned about the security of my data. Will those using Nexus DB have access to my project? Oh, this is another good question. Because people often, sometimes, I don't know why, but I do mention this a few times. So first of all, if you access Nexus DB, there is no virus or something that's going to suck the data out of your project, so you don't have to worry about that. You have to have an active means to submit your project. So you have to click Submit Project and then go through a couple of screens to move your data to Nexus DB. Once the data goes into Nexus DB, it goes into under My Projects, and nobody will be able to access it unless you give them right to. So you see these projects that I have, like this very old project, there's no group that has access, so nobody can see that. But if I want to allow somebody else to look at it, I have to change the permission, and you can see it's not visible to anybody. But if I want my colleagues at the company to see it, I can make them just visible or visible and downloadable, and then they can get access to it. So what we've done with the TCGA data is we have made that available to anybody, but public, which means anybody with a Nexus license can access it. So thanks for that. Let me see. We really need to get level 1 data. I agree. Getting level 1 data is a bit challenging because you have to write a project and justification for. And we have done that and been approved for getting the GBM data. We're working for the additional set. Okay, so at this time, I would like to, we can continue with questions, and thank you for the questions. They're great feedback. But we have a few poll questions to appreciate getting your feedback.